Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 for October 2023. Let's begin with the first question. In question 1, the structure of a chloroplast is related to its role in photosynthesis. The diagram shows a chloroplast. So this is the stroma. We see the starch grain, the ribosome, the outer membrane, DNA, inner membrane, as well as the thylakoid membrane. They say which is the site for photolysis. It occurs in the thylakoid membrane, so the answer should be T, which is C. And next I say which contains a gene coding for rubisco. Because they talked about genes, this is going to be DNA and it's going to be part V, which is going to be D here. Next they say where do hydrogen ions accumulate for use in photosynthesis? It occurs inside the thylakoid, which is going to be structure S, so the answer is C. And in D, how many of the structures labeled P, T, U, and W are made of phospholipids? Remember, P is the ribosome, no phospholipids. T, thylakoid membranes. Actually, these are all membranes, so they have phospholipids. So the answer is going to be 3 of them. Next, they say which describes a phospholipid. A phospholipid has 1 glycerol, 2 fatty acids, and a phosphate. So a glycerol attached to 2 fatty acids and 1 phosphate group, and A is going to be the answer. In part E, the length of a cell was measured. It was found to be 0.0085 centimeters long. The ratio of the length of this cell to the length of the chloroplast is 11.7 to 1. Calculate the length of the chloroplast. So if we see cell wall to chloroplast, that is the ratio, is going to be 11.7 to 1. Yet we know the actual length of this, which is 0.0085. So this is going to be 0.085 divided by 11.7. I'm trying to get the value of x. And that gave me 7.265 times 10 to the power negative 4 centimeters. But they want the answer in micrometer. So that is going to be 7.265 micrometers since 1 centimeter is 10 to the power 4 micrometer. So the answer is 7.3 micrometer. So this brings us to the end of question 1. Let's continue to question 2. In question 2, the body responds to infection with non-specific responses and the immune response. Inflammation is characterized by heat, redness, and 2 other characteristics. Name the 2 other characteristics. This is going to be pain as well as swelling. The next is to explain how heat helps the body respond to infection. Heat is going to increase the temperature, making the immune cells to work better, but also it's going to prevent the increase in the number of the pathogens because it's going to affect how their enzymes work. So I said heat increases the temperature, providing ideal conditions for faster phagocytosis. The increased temperature destroys the enzymes present in pathogens, preventing their reproduction. In part B, describe how interferon helps the body respond to infection. Interferons are a body's response against viral infections. So these are going to be secreted by cells that are infected, and this is sometimes useful in triggering the T killer cells to destroy cells that have been infected. So I say they are secreted by the body cells infected with viruses against viral replication to slow down the rate of viral protein synthesis. Interferons pass out of the infected cells, stimulating the T killer cell activity. In part C, the immune response results in the production of antibodies following the first infection with a pathogen and no further infection with the same pathogen. The graph shows the concentration of antibody in blood plasma following the first and second infection with the same pathogen. So on the vertical axis, we have the concentration of antibody in blood plasma, and on the horizontal axis, we have time. We see the initial infection and then secondary infection. Initially, the concentration takes longer to increase, and the highest concentration reached is going to be well lower than what occurs in second infection. Second infection, we see it takes a shorter period of time, and we have more concentration, so it's faster, and it's going to produce more antibodies. So they say compare and contrast the changes in concentration of antibody in the blood plasma following the first infection and second infection. I said in both, an increase followed by a decrease in the concentration of antibodies occurs. After first infection, the concentration of antibodies takes longer to peak and the maximum reached concentration is lower. However, after the second infection, the concentration of the antibodies is going to be higher and it's going to be produced at a faster rate. So this brings us to the end of question 2. Let's continue to question 3. In question 3, the growth of bacteria in a liquid culture was investigated over a 14-hour period using an optical method, which is turbidity. A calibration curve was used to convert the turbidity readings into the number of bacteria per centimeter cubed of liquid culture. The graph shows the calibration curve used. So here we have the number of bacteria per centimeter cubed and then turbidity. That was the curve given to us. They say the table shows the results of the first 10 hours of the culture. We can see as time increases, there is an increase in turbidity, but after it reaches 0.8, it does not change, meaning there is no more increase in the number of bacteria because we can see the increase becomes 0. Yet initially we saw a big increase in the number of bacteria. So they say complete the table to show the increase in cell number between 4 to 6 hours of the culture. At 4 hours, we can see we have 0.04, which is going to be this one here. And the number of bacteria is 6.0 times 10 power 4. The next one is at 6 hours where we have 0.56. That is a turbidity. At 0.56, I got 9.1 times 10 power 5. So here my answer, which is going to be the increase, is 8.5 times 10 power 5. Remember again, at 4 or at 0.04, we have 6.0 times 10 power 4. The change here to give us a 9.1 times 10 power 5, the change is going to be 8.5 times 10 power 5. So that's the answer. Next, they say explain the changes during the first 6 hours of this culture. If I take you back, during the first 6 hours of the culture, initially we had no increase, but after time we saw some increase. This is because initially the culture or the microorganisms were getting used to the conditions, and afterwards they were used to the conditions like temperature and pH, and it was easier for them to increase in number. So I said in the first 2 hours there is no change in the number of bacteria because the bacterial cells are adjusting to the conditions, temperature and pH. After that, there is an increase in the number of bacterial cells because the conditions, which are temperature, pH, nutrient, oxygen levels, and so on, are optimum for growth. In part C, the number of cells remained constant after 8 hours to the end of the 14-hour period of this culture. Bacterial growth was also investigated using dilution plating and colony counts. Describe the method used in dilution plating. Usually in dilution plating, they carry out serial dilution, which is accompanied by plating or spreading some of the volume from the dilutions onto petri dishes, and then they culture for some period of time and count the number of colonies that can be distinguishable in order to know the concentration. Next, they say the sketch shows the shape of the growth curve produced using dilution plating and colony counts between 8 to 14 hours of this culture. So we can see they're constant and then they begin to decrease because there is more death than the number of new bacterial cells that are produced. So they say there are differences in the numbers of bacteria between 8 to 14 hours, measured using optical methods, which is turbidity, and measured using dilution plating and colony counts. One thing we need to know is the turbidity method cannot distinguish between those that are dead and those that are alive because it passes light through the sample, and those that are dead are also going to cause blacking of light, so it will not distinguish them, while dilution accompanied by colony counts only counts those that are alive, since those that are alive are the ones that are going to grow into colonies. So we're going to have different results. We expect the number of estimates of bacteria to be more in the turbidity method than in this other method of plating and counting the colonies. So I said dilution plating only measures living cells, while using turbidity method considers the total number of cells present. In the dilution plating method, between 8 to 12 hours, the number of bacterial cells was constant since the number of new cells and the dead cells was equal. In the turbidity method, the numbers remained constant because no new cells were produced. Between 12 to 14 hours in dilution plating, the number of cells decreased since death occurred, but no more cells were produced. So this brings us to the end of question 3. Let's continue to question 4. In question 4, human breast milk supports the growth and development of a baby. One important component is a group of carbohydrates called oligosaccharides. These contain between 3 to 10 monosaccharide units. During the first few days after birth, the milk produced is called colostrum. By 2 weeks, the composition of milk has changed and it's called mature breast milk. The chart shows some of the nutritional components of colostrum and mature breast milk. So we can see the components. They say estimate the percentage composition of oligosaccharides in the colostrum. Now this question is talking about only estimations, but I was able to get this protractor here. And we can see if we measure the angle. This is about 36 degrees. If you can see this angle here, around here like that, it's about 36 degrees. If we add it to 180, which is that, we get about 216 degrees. And again, this is just an estimate and the answer is going to be varied, but I just wanted to be kind of sure. In the exam, you can just estimate. Even if you took this to be 30 degrees, it will be fine. So I used the actual one and I get about 36 degrees. So 36 plus 180, you get 216. The total angle, because this is around the circle, that is going to be 360. So 216 divided by 360 times 100 gives you the portion of oligosaccharides, which is going to be 60%. In part B, oligosaccharides in breast milk have been shown to be important in establishing a healthy gut flora in the developing baby. The effect on the types of bacteria in the guts of babies fed on breast milk or another source of milk was investigated. The graph shows the proportion of 5 types of bacteria in the 2 groups of babies. So here we see the different bacteria. We can see the groups fed with breast milk and the group fed with another source of milk. We see we have a higher concentration of bifidobacterium for both. And then we see for the group fed with breast milk, we're going to have the least in these ones here, where the groups fed with another source of milk, we're going to have the least in this one here. So they say describe 2 conclusions that can be made about the gut flora of babies fed these 2 types of milk. So I say they all have the same types of gut flora, but in different proportions. In both, bifidobacterium was present in the highest proportion. Babies fed on breast milk had the least Veillonella, while those fed on other milk sources had the least bacteroides. Next, I say explain the importance of healthy gut flora in preventing infection in a developing baby. Healthy gut flora outcompetes or tries to compete for space and nutrients with pathogens. So if we have enough healthy gut flora, the pathogens are going to be outcompeted. So I said healthy gut flora competes for space and nutrients with pathogens, and this prevents pathogenic bacteria from colonizing the gut. In part C, Group B Streptococcus, GBS bacteria are a common cause of infection in babies. These bacteria are becoming increasingly resistant to antibiotics. Scientists have shown that oligosaccharides isolated from breast milk have antimicrobial activity against GBS bacteria. Explain the possible advantages of using these oligosaccharides instead of antibiotics to treat GBS bacteria infections. Number 1, these come from milk, so they are safe, meaning they are not going to cause selection pressure, which could lead to resistance like we see with antibiotics. And number 2, they're going to encourage the growth of good bacteria. So I said using oligosaccharides increases the number of gut bacteria. These outcompete the GBS bacteria, decreasing their number. Using oligosaccharides decreases the use of antibiotics. So fewer GBS are exposed to the antibiotics, which are going to be selection pressure, and so resistance can be overcome. Also, since these oligosaccharides come from breast milk, they have fewer side effects. So this brings us to the end of question 4. Let's continue to question 5. In question 5, cultivation of seaweed for food is well established in Asia. In 2017, the global harvest of seaweed was 32.9 million tons. Of this, 96.6% was cultivated. Calculate the mass of cultivated seaweed and give your answer in standard form. So this is going to be 96.6 divided by 100 times the total, which is 32.9 times 10 power 6. and I got 31781400, which is 3.18 times 10 power 7 tons. And next they say seaweeds are photosynthetic organisms. Photosynthesis can be described as a reaction requiring light to split apart the strong bonds in water molecules, storing the hydrogen in a fuel by combining it with carbon dioxide and releasing oxygen into the atmosphere. Which type of bond is broken in the water molecule? This is going to be the covalent bond. So the answer is A, which is a fuel that stores hydrogen in plants and seaweeds. That is going to be glucose. Which row of the table shows the reactions that combine hydrogen with carbon dioxide and release oxygen into the atmosphere? Hydrogen is combined with carbon dioxide in the light-independent reactions, and then oxygen is released into the atmosphere in the light-dependent reactions, so the answer should be C. Next, they say in Europe, cultivation of seaweed takes place on a small scale. Large-scale cultivation of seaweed is now being considered. Effective management of the conflict between human needs and conservation is necessary before large-scale cultivation can be used. The diagram shows seaweeds grown by suspended cultivation, a method used in Asia. So here they say rows of seaweed suspended on wires in the sea. They say the diagram shows some of the factors that have to be considered when managing the conflict between human needs and conservation. So the absorption of light. We know if we cultivate more seaweed, there is going to be more sunlight that is absorbed, and that is going to increase the carbon fixation as well as the numbers of seaweeds that grow. Labor. This is going to be a source of income for some people. They're going to be hired in order to work and they'll be paid to earn a living. Absorption of carbon dioxide. Cultivating seaweed is going to decrease the CO2 concentration in the atmosphere. Noise. We saw they use machinery, and then the people who are going to be working on the seaweed, the noise pollution could affect the organisms that live in the water. The artificial habitat, this could be both advantageous and not, but the seaweeds that are grown are going to be or act as an artificial habitat, which allows for organisms to thrive. However, some organisms would come and thrive in there and destabilize the habitat of that area, disorganizing the food chain and so on. Transport and processing, again using machinery, maybe requiring transportation methods. This could be disadvantageous because it's going to lead to increase in fossil fuels being used and so on. So that could lead to increase in CO2 concentration. So they say discuss the considerations that need to be taken into account when deciding whether or not large-scale seaweed production is suitable in Europe. Use the information in the two diagrams and your own knowledge to support your answer. Like I said already, here I looked at it in two different sections. I began with the first diagram and then with the second one. So from diagram 1, seaweed is suspended on wires in the sea. Some organisms can be hooked on the wires and die. Also, the suspended seaweed takes up space that could be occupied by other organisms. The suspended seaweed could act as a habitat for organisms that could alter the water ecosystem. This depends on the organisms that are inhabiting the ecosystem. If they are predators or they are prey, there is a way they're going to affect or have an effect on the food chain and therefore alter the ecosystem. From diagram 2, presence of seaweed creates a habitat. It absorbs sunlight and carbon dioxide during photosynthesis, provides a source of food, and this increases biodiversity of the water ecosystem. Seaweed cultivation provides labor, giving a source of income to the locals or the people who are employed. However, the noise created by the workers and machinery could cause stress or could stress the organisms. Seaweed habitat is artificial, and once removed, the food chain is destabilized and organisms could die. Also, the use of machinery could pollute the water, decreasing biodiversity. The workers could also introduce diseases that could affect the organisms. So this brings us to the end of question 5. Let's continue to question 6. In question 6, telomere is a repetitive nucleotide sequence at the end of a chromosome. The telomere protects the end of each chromosome from damage. Each time DNA is synthesized, the telomeres become shorter. When telomeres become too short, the cell dies. The photograph shows telomeres on the end of chromosomes. They say which diagram shows a mononucleotide of DNA. Here the answer should be C. This is the one that is arranged in an orderly way. So that is the mononucleotide. The others do not make sense because the bonds are not positioned where they should be for some of the components within the structures. Next says name the bond that joins nucleotides together in a strand of DNA. It is a phosphodiester bond. And then the photograph shows a common lizard, which is that lizards cannot regulate their body temperature and are at risk of extinction due to global warming. The effect of temperature on the length of young lizards was investigated. The change in abundance of lizards of all ages over a 12-year period was also investigated. There was a positive correlation between temperature and length of young lizards. This could be due to enzyme activity. Explain the correlation. So of course, because it's enzyme related, the higher the temperature, the longer the lizards. This is because increasing temperature increases kinetic energy of the enzymes and the substrate, so there will be more successful collisions between the enzymes and the substrate. So the enzymes include DNA polymerase. These are involved in replication of DNA, RNA polymerase involved in formation of messenger RNA that is going to later be involved in protein synthesis. So more proteins are going to be made, more DNA is going to be made, more mitosis is going to occur, and there is going to be an increase in length. So I said, and the substrate like nucleotides to achieve faster DNA replication, faster mitosis, faster protein synthesis for faster increase in length. Then they say the scientists used the data to predict a relationship between the length of the young lizards and the percentage change in abundance of lizards over the 12-year period. The graph shows this relationship. So we can see on the vertical axis we have the length of lizards, and down here the percentage change in abundance. They say describe the relationship between percentage change in abundance of lizards and length of young lizards. If the length is higher, until about 46, we can see the values are in the negatives. So there is going to be a decrease in the percentage abundance, meaning lizards that are longer are not going to be abundant. Between 46 and about 45.5, we can see there is an increase in the abundance. So meaning this length, between this length here, there is an increase in abundance, and as the length increases, there is a decrease in abundance. So I say the length of the lizard affects its abundance, above 46 millimeters of lizard length. As the length increases, the percentage change in abundance decreases, and between 45.5 millimeters to 46 millimeter length, the percentage change in abundance increases. Then they say the scientists also looked at the length of the telomeres in the cells of these young lizards. Explain why the abundance of the longer lizards is affected by the number of cell divisions taking place as they grow. Use the information in the question to support your answer. Remember we said for lizards to be longer, there is going to be more mitosis, and that will require more DNA replication, meaning the telomeres are going to become shorter and shorter. There is going to be more cell death, meaning more lizards could die before they reach maturity, and that's why maybe we see fewer longer lizards. So I said longer lizards would have gone through more cell divisions, so more DNA synthesis would be required. The telomeres will be shorter, leading to more cell death, so they die before they are longer, decreasing their abundance. So this brings us to the end of question 6. Let's continue to question 7. In question 7, cancer immunotherapy stimulates and strengthens an individual's active and passive immune systems to destroy cancer cells. The diagram shows how different types of cancer immunotherapy can be classified. So we have cancer immunotherapy, there is active immunotherapy, passive immunotherapy. The active immunotherapy involves anti-cancer vaccines, and the passive ones, there is monoclonal antibodies, adoptive cell therapy, and oncolytic virotherapy. This suggests what the difference is between active immunotherapy and passive immunotherapy. Of course, in active, there is going to be formation of memory cells because the immune system is going to be activated to produce antibodies. But in passive, the antibodies are going to be given. So I said active immunotherapy activates the immune system, leading to production of antibodies and memory cells, while passive immunotherapy only involves receiving antibodies. In part B, cancer cells result from mutations Some of these mutations can result in the formation of altered membrane proteins called neoantigens. The neoantigens are not present on the surface of normal cells. Explain how a mutation can result in the formation of neoantigens. Now, these neoantigens are proteins, so if there is a mutation in the DNA, that is going to be transcribed into messenger RNA, which is going to be used to form a polypeptide chain. That polypeptide chain is going to have different amino acids at different locations, changing or altering the protein produced. So I said mutations change the base sequence of DNA, so a wrong or wrong messenger RNA codons lead to formation of a faulty amino acid sequence, so a wrong protein structure is going to be formed. So this is some active anti-cancer vaccines can contain the messenger RNA that codes for neoantigen. Explain how vaccines containing the messenger RNA coding for neoantigen stimulate the immune system. This messenger RNA is going to be used to produce the protein, which is the neoantigen. In presence of this neoantigen is going to trigger the immune system in order to produce memory cells as well as antibodies against the neoantigens. So I said the messenger RNA is translated into the neoantigen, which is the protein, and these are recognized by the immune system as non-self molecules. The macrophages will carry out phagocytosis and antigen presentation to the helper T cells. The T helper cells produce cytokines stimulating B cells and T killer cells, so any cancerous cells could be destroyed. Next, they say monoclonal antibodies are made in a laboratory. Large numbers of identical antibodies specific to one type of antigen can be made. This is very important, larger and then specific. They say explain how these antibodies help the immune system to destroy cancer cells. Antibodies carry out opsonization, they cause agglutination, and so on. So I say they bind to the neoantigens or marking or opsonizing them so that phagocytosis occurs faster. And they also cause agglutination of cancer cells so that more are engulfed. In adoptive cell therapy, T cells are taken from the patient and large numbers of genetically identical cells are made in the laboratory. These cells are then injected back into the patient. Explain why it's necessary to produce large numbers of genetically identical cells to make this treatment effective. So there should be large numbers because we need more T cells or T helper cells to stimulate the B cells as well as the T killer cells into action. And we need them to be genetically identical so that they can cause the same effect, meaning to prevent rejection in the body. So I said large numbers of T helper cells are required for activation of more B cells and T killer cells. The cells must be genetically identical to prevent rejection. In part E, in oncolytic virotherapy, viruses that destroy cancer cells are injected into the patient. Not all types of virus are suitable for this therapy. Suggest 2 features of a virus that make it suitable for use in this type of therapy. The virus should not be pathogenic. Number 1, it must be able to destroy the cancer cells. It should have antigens or molecules that are complementary to the cancer cell neoantigens. So this brings us to the end of question 7. Let's continue to question 8. In question 8, the trophic level of an organism is the position it occupies in a food chain. The diagram shows the energy flow between trophic levels of a food chain. So this is what we have here. They've given us the energy available to the next trophic level and energy entering the trophic level. So let's see the first question. They say, calculate the net primary productivity as the percentage of the gross primary productivity in this food chain. This is going to be the gross primary productivity, and that is the net primary productivity, meaning whatever leaves the trophic level 1 to go to the next trophic level. So the difference between the two is going to be what goes into respiration, which is that. So this is going to be the net primary productivity divided by the GPP times 100, giving us 42.6%, and that is the answer. The next they say complete the right-hand side of the diagram by adding arrows of the missing information in the box. So here, the difference between that and that is going to be that. Then the difference between that and that is going to be that. The difference between that and that is going to be that. And finally, the difference between that and that is that. So adding everything up, I got 56672 kilojoules as my answer. And next is explain the importance of enzymes produced by microorganisms in decomposition. These enzymes are released, they carry out hydrolytic reactions, breaking down the bigger molecules into smaller substances that are soluble so that they can either be absorbed by the microorganisms or some could leach into the soil. So I said enzymes carry out hydrolysis of biological molecules in the decomposing substances into smaller soluble molecules. These molecules can be absorbed by the microorganisms for respiration, or they can be added to the soil as part of the carbon cycle. In part B, some animals eat a diet of meat and plant material. These animals can be given a trophic level, number which is TLN, based on a scale of 1 to 5. Animals that eat only plants have a TLN of 1, and animals that eat only meat are at the top of the food chain have a TLN of 5. Humans are an example of animals that eat both plants and animals. The graph shows the change in the TLN for the total population of China and India from 1960 to 2010. So we see trophic level number on the vertical axis and then the years. Over time, we see there is an increase in the TLN number, meaning people are choosing to have meat more than previously. As time goes on, they're eating more meat. So this explains what the information in the graph indicates about the deaths of the population during this time period. I said overall, as time increases, the TLN number also increases. More people incorporated or are incorporating meat in their diet. Before 1983, the proportion of meat was less and just fluctuating. We can see up and down, fluctuating. After 1983, there was a big increase in the proportion of meat in the diet. We can see that big jump. From the graph, people in these countries still have a high proportion of plant material because the TL number is still below the average, which is 2.5. So this brings us to the end of question 8. Let's continue to question 9. In question 9, the photograph shows a sea turtle, which is this one here. They say sea turtles can take in plastic debris when they eat. This plastic can harm the turtle. The graph shows the global production of plastic per year from 1950 to 2015. We can see as the time increases, there is an increase in the global production of plastic. There is a general increase. They say calculate the mean rate of global plastic production from 2000 to 2015. This is about 15 years, so I need to get the difference, 2000 to 2015. In 2000, we can see it's about 210 times 10 power 6 tons. And then here in 2015, we can see it's 380 times 10 power 6 tons. So the difference is going to be 380 minus 210. Then to find the mean rate is going to be the difference divided by 15, which is 170 over 15, giving us 11.33. Next, they say estimate the global plastic production in 2023. Now remember, this is per year, and if I go back here, this is until 2015 where we have 380. So I'm gonna kind of extrapolate or go ahead from 2015 all the way to 2023. That is about 8 years. Now until here we have the average or the mean rate was 11.3. So 11.3 times 8 years, and then I'll add it to the 380 to find an estimate. So I said at a rate of 11.3 times 10 power 6 tons per year, for the 8 years, which is between 2015 to 2023, it's going to be 11.3 times 10 power 6 times the 8 years, giving us that. This is the estimate that can be produced in the 8 years. Now to find the total, which is going to be that produced until 2015 plus that produced in the 8 years until 2023, that gave me 470.4 times 10 power 6. Writing this in million tons, it's 470.4 million tons. So that's my answer. And next I suggest why this value may be an understatement. Use the information in the graph to support your answer. If we go back to the graph, there are those fluctuations that can occur. So what if other fluctuations occur and the rate is not going to be continuously 11.3 million tons? Then the value is going to be understated. It could be up or down. So here I say the graph shows fluctuations, so the production of plastic is not linear, meaning there is a chance that fluctuations could occur between 2015 to 2023, and alter the estimate that we have predicted. In Part B, one group of scientists determined the number of pieces of plastic in the guts of in vitro seed taros. They then modeled this data to estimate the probability of death due to plastics. The graph shows the results of this modeling. On the vertical axis, we have probability of death due to plastics, and on the horizontal axis, we have the number of pieces of plastic in the gut. This is similar to a sigmoid shape, meaning initially, as the number of plastic pieces are fewer, there is less death or less probability of death. And as we reach a certain number, we see there is an increase. And over time, it kind of levels off as well. Describe how the probability of death due to plastic increases with the number of pieces of plastic in the gut. This is just describing what we see here. So I said as the number of pieces of plastic in the gut increase, the probability of death due to plastic increases. The increase shows a sigmoid shape. Initially, the probability is 0. However, after a certain number of plastic, the probability of death due to plastic increases drastically. Next, I say describe two limitations of using this graph to determine the probability of death due to ingestion of plastics. From the data, we are not given a lot of information. For example, we do not know the size of the plastic. We do not know the size of the organisms, the type of plastics, and so on. So I say the graph did not consider the size of the plastic. There is no data for how long the plastic was inside the taro. There is no information about the taro size as well as age. And some taros could have plastic in their guts, but die due to other unrelated conditions or conditions unrelated to plastic. In Part C, mangoes are tropical fruits that are harvested before they are ripe. Plastic packaging is used to wrap mangoes after they have been picked. This preserves the fruit during transportation and storage. Banana stalks have a high content of cellulose and have been modified to use as packaging material for mangoes. A study compared the use of these two types of material and their decomposition. The graphs and the table show the results of this study. So the first graph, we see the percentage of mangoes that are diseased. There are more diseased mangoes in plastic, which is going to be the highest here. And then the lowest is going to be in the modified banana stems. Then here we see percentage of total mass of mangoes lost. There is greater loss in those that are not enclosed in packaging. And there is going to be the least loss in plastic. Then down here we see the appearance of the packaging. They say time of decomposition. This is about biodegradability, the time of decomposition of the package. For plastic, we can see it's still there and almost little has changed over a period, but for banana, we can see after 4 weeks, there is a lot that has decomposed. So they say discuss the advantages of using packaging made from modified banana stalk. Use the information in the graph, the table, and your own knowledge to support your answer. These are going to be biodegradable. They're going to be sustainable. They're not going to create any toxicity. They maintain fair enough properties of the mangoes or integrity of the mangoes. We can see the percentage of mangoes that are diseased are going to be lower. We also see the total mass loss is going to be less, and so on. So I say it from graph 1, using modified banana stem packaging leads to a lower percentage of diseased mangoes, so more mangoes will be consumed, providing a source of food and a source of income. From graph 2, modified banana stems result in lower percentage of total mass loss. Plastic packaging led to the least loss in mass. Packaging that decreased loss in total mass decreased the rate of dehydration. So mangoes with good water content are helpful in providing dietary water. From the table, greater decomposition occurred for the modified banana stem packaging than plastic because bananas are biodegradable while plastics are not. Decomposed banana stem packages are carbon neutral. They are inexpensive, they are sustainable, and they do not lead to release of toxic gases like some plastics. Lastly, planting bananas decreases the atmospheric carbon dioxide levels through photosynthesis. So this brings us to the end of question 9 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.